All right, this is Scott and Kristen, and we're going to go over week three's genetics problems, going over epigenetics and sex-related inheritance.
All right, so in question 1, it talks about Angelman syndrome, and the imprints is silent, or the paternal allele is silenced. So we're crossing a heterozygous female and a heterozygous male, and so what we did for this one was just put squares again to get the correct answer, which there's half non-diseased and half diseased. And then part B, it talks about a heterozygous male and then a homozygous recessive female. And we went through that same process again and got the genotype is half heterozygous and half recessive, and then all of them are going to have Angelman syndrome.
Cool, thank you. So the next question asks about a woman that has triple X syndrome, which means that she has 3 X chromosomes, and it's asking about the XX, the molecules XX, which are on the X chromosome, and they've been deleted from two of the X chromosomes in each of her cell. So it's asking about how many bar bodies are going to be found inside of this cell. The answer is that there's no bar bodies. There has to be at least 2 XX, at least 2 of them, for the X chromosome to be inactivated. So because there is only one, because there aren't 2 of them, there's going to be no bar bodies. So the question D asks about isolating a cell from a female cat with 2 X chromosomes, and the T6 genes on each X chromosome are non-functional, while the X-ist genes on each X chromosome are functional, and it's asking about how many bar bodies are expected to be found in the cell. Aside from butchering the reading of that question, I'll try to write it out on the board. So it says SIX, and then there's TXIS. Right. There we go. Okay. X.
Oh no, that one's right.
6, and then T6X. Okay, there we go. So with these 2 ones, the T6, so this is going to prevent X inactivation. So it's going to make sure that the X inactivation doesn't happen, and then this is going to cause X inactivation, and so that means that the X is going to be inactivated. And so in this question, the X-ist gene is functional, this one here, and the T6 is non-functional. And so since this is non-functional, then all of them are going to be inactivated here. And so the answer is there's going to be 2 bar bodies, 1 on each X chromosome.
Okay, then for question 2, fill in the blanks. Mostly we just found those from our notes also. So A is the SRY, B is MIC2, C is one dwarfed, one female for the phenotype of the offspring. D is sex-influenced inheritance. E is genomic imprinting and dosage compensation. And then F, the two molecules that form halves of a double-stranded RNA is going to be the X cis and the T cis, which we just talked about in the last question. And then G is going to be autosomal chromosomes, and then X-linked chromosomes.
Cool. So the next question is asking about X-linked recessive disorder called I'm not even going to try to say that.
OTD.
OTD. Yeah, that's the easy way to say it. So the woman's mother is homozygous and unaffected, and the woman's father has this disease. And so it's asking what the genotype of the woman's father is and the genotype of the woman's mother. And so the father, he's going to be homozygous recessive. He's going to have an X and a Y, but her dad is going to have the recessive allele on his X. And then the mother is going to be homozygous dominant, meaning that she's going to have two X chromosomes, and neither of them are going to have OTD on them.
Okay, and then it says if a woman has children with a man who does not have OTD, what is the chance of their first child being son? So for this, we did a little Punnett square with the mom, which is going to be, she's going to be heterozygous, and then the dad, which is going to be, and then we found out that it's going to have a 25% chance of the first son to have OTD.
Yep. Exactly. So the next question is still talking about OTD, and it says, identify the genotype of a male with whom the woman could produce a daughter with OTD. And so what we did for this one is let's see, this is 3, question C. So we took basically we like reverse engineered a Punnett square and tried to identify how you could create a daughter with OTD. And the only way that we could do this is if we had her mate with a man who has OTD, and he would have to be homozygous recessive for the disorder.
And then for part D, it asks for the different genotypes and phenotypes of an offspring from part C. So you're going to get a little bit of everything. So you'll get heterozygous female, which will have no OTD. And then you'll get a recessive female, which will have OTD. And then you'll get an unaffected male, so no OTD. And then you'll get a male that does have OTD. So a little bit of everything.
Exactly. Cool. So now we go on to question 4, which asks about a autosomal gene that is dominant in both males and females. Dominant in males and recessive in females. Totally different. And so it gives you a situation where you have a cross where the progeny of two different goats, between parents, is 3 beard and 1 no beard, and that's just among the males. And so it asks what the phenotypes of the parents are, and since it's 3 to 1 for the men, And for men, this is going to be a homozygous, sorry, this is going to be a dominant gene. Then that means both of the parents are heterozygotes. The father has a beard and the mother has no beard.
Yep. And then for part B, it asks you about females this time, and the ratio is 1 to 3, 1 being with a beard, 3 being not with a beard. And it would be the same as part A. Both have to be heterozygous. The father has a beard and the mother has no beard.
Yep, exactly. All right, and now it's asking about a cross between a male with a beard and a female without a beard, and they produce a 1-to-1 beard to no beard ratio among the females. And so half of the females are going to have a beard, and half of them are going to have no beard. And so this is kind of like this is kind of to get half of something, you'd have to have one parent that is homozygous dominant, and then the other one that's heterozygous, or one is homozygous one way and the other is heterozygous. In this case, the father is homozygous dominant and the mother is heterozygous. So that's how we got the answer to that one.
Okay, and then in part D, it says both the male and females lack beards, and they produce a 1-to-1 ratio of beard and no beard among males. So what are the genotypes of the parents? So we reversed engineered that too with the Punnett square, and we found that the father is homozygous recessive and the mom is going to be heterozygous.
Exactly. Cool, and now we move on to the last section here, part 5, and it's asking about X-linked inheritance and reciprocal crosses. And this one was pretty straightforward. First question was asking about cats. Orange is determined by the B allele, and black coat color is determined by, they're both determined by the B allele. Orange is by the recessive, and black is by the dominant allele. The heterozygous condition of big B, little b produces a phenotype known as tortoiseshell. And so it's asking about the genotypes and phenotypes if you cross an orange cat with a tortoiseshell cat, orange male and tortoiseshell female. So we ended up finding out that the genotypes for this one was a little bit of everything. You get a female cat that's orange, a female cat that's tortoiseshell, a black cat that's male, and one orange male.
Yep. And then part B asks, what is the genotype of a male tortoiseshell cat? And this is the same as Klinefelter's disease in humans, not in cats though, I don't think.
Yeah.
And then so the male would have to have 2 X chromosomes and have them be heterozygous, and then there's the Y chromosome still there.
Mm-hmm. Each one of those X chromosomes would have to have a different allele, like she showed you on them. Next one is asking about fruit flies. The next 2 questions are asking about fruit flies. And there's a mutation that caused scalloped wings. And so scalloped wings are the result of an X-linked recessive mutation. And it gives us some, it asks about what the genotypes and phenotypes of the progeny would be when the reciprocal crosses are done. So the first one, if we took a cross where the male has scalloped wings, so he would have an X with a scalloped wing and a Y chromosome, and the mother would have 2 normal. Then you cross them and you end up getting all males have the XY, and females have, all males have normal wings, and all the women would be carriers with normal wings. And the women would be the only ones that get the scalloped wings allele. And then the second reciprocal cross is the opposite. The woman has scalloped wings now. And in this situation, you would have all of the offspring be heterozygous due to the scalloped genes allele or scalloped wings allele would end up being on each one of the progeny.
All right, and then part D is related to part C, and it asks you to indicate the genotype and phenotype of the progeny produced when reciprocal crosses are done. And so for this, they're all going to be normal looking, or normal winged, I guess, and they're all going to be heterozygous. And that concludes our video. Thanks for watching, guys.
Yep.